Welcome back, Year 12s, to our next video looking at the electron transport chain today. So we looked at the Krebs cycle last class. Essentially, it helps create NADH and FADH2, which are used as electron carriers, and then of course hydrogen carriers, which are protons. All right. Also, carbon dioxide is released. ATP is created, 2 molecules in that case. All right, and you can see that here. Alrighty, so let's look at the electron transport chain in this video. All right, again, be aware of this 26 or 28 ATP. It's not 27, it's not 25, not 29. It is 26 or 28 from the electron transport chain, totaling to be 30 or 32 ATP for all of aerobic respiration. And of course, fermentation there is 2 ATP. All right, the electron transport chain. So it's the third process of cellular respiration. Okay, and we should be aware that it's located on the folds of the inner membrane there, so the cristae of the mitochondria. Alrighty, so again, we have our double membrane of the mitochondrion. All right. It's going to be happening on the folds here. All right, all of these little parts, that's where the ETC occurs. All right, beautiful. So energy from the electrons unloaded by NADH and FADH2, because that's their primary purpose. All right, allows hydrogens to flow through the plasma membrane and create this proton gradient. Remember, a proton is just hydrogen ion, so H plus in this case. Okay, and that drives significant ATP production. Obviously, the highest ATP production, as we just mentioned, 26 or 28, compared to that of 2 in glycolysis and 2 in the Krebs cycle. All right, the electron transport chain is where a majority of the ATP is produced. Yep, it also converts NADH and FADH2 back to their original NAD plus and FAD forms, which means they can be used in glycolysis again, and then the Krebs cycle again. Okay, so yeah, it is important for those things too. All right, just being aware of ATP synthase here, that enzyme that's essentially, it's just sitting in that plasma membrane, which is the inner mitochondrial membrane. Okay, and it is a channel protein, so the hydrogen ions will flow through it passively. Okay, and due to the action of many other things like the electrons and oxygen coming along, all right, there's a concentration gradient of hydrogen ions. All right, and again, just to reiterate what that is, if this is our membrane, all right, we have lots of hydrogen ions up the top. All right, and we have less down the bottom. That is what we call a concentration gradient, because it's high at one spot, low at another. So then, if there is a channel protein, these ions will passively diffuse through, okay? Through simple diffusion. So again, H plus is positively charged. So if it has a plus, it's positively charged, which means it's missing an electron. Okay, so this is where a lot of students get confused. Again, this is a bit of a nuanced point, but you need to be aware of it. Okay, so if something has a plus, it means it's positively charged. Okay, and how does that work? That means, has it gained an electron or lost an electron? The point is, it's lost an electron here. Okay, because the electron is inherently negative. So electrons are negatively charged. Okay, so if you take an electron away, all right, as you can see in the image here, hydrogen with 1 electron in that shell. Now it's hydrogen with no electrons in that shell. You've taken away an electron. All right, so you've taken away a negatively charged molecule. So now this is a less negative. In other words, it's positive. All right. So that's what a proton is, H plus. It's just a hydrogen molecule missing an electron. All right, so this is now our electron transport chain. There's a lot going on here, okay? Again, biochemical pathway is not required, but understanding this does allow you to, have a good understanding of what is going on, okay? So here we go. All right, we have, of course, our beautiful NADH and FADH2, and they're working their magic with these electrons here. Okay, the electrons go through the membrane, through these proteins here, again, of which we don't need to really know what they are. All right, but the point is that these proteins start to create that hydrogen plus, or the proton gradient. Okay, because these protons, so hydrogen plus, start to go through these. They start to flow through, all right, due to the electrons. All right, so as you can see, we have a hell of a lot of these hydrogen ions up here, and we have a few on this side down here, okay? So that means that's a really strong gradient. All right, then if we look over here, we have ATP synthase, all right? And as you can see, the hydrogens in mass up here, so they're going to passively diffuse through the channel protein, of which ATP synthase is. As they diffuse this through this bottom part here, it actually spins. The enzyme spins, the protein spins. It's incredible. And as it spins, it flicks an inorganic phosphate here. It flicks it onto the ADP, boom, which then creates your ATP, because ADP is diphosphate, ATP is triphosphate. Okay, so plus one phosphate for 2 equals 3, yeah, ATP. Okay, and that happens repeatedly, all right? So realistically, the hydrogen will be sitting up here, it'll passively diffuse down here, and it will float around here. All right, it might get attached to an oxygen molecule, all right, as well. It might get attached to an oxygen molecule, making water, H2O, okay, or it continues to float around and eventually will go back up, okay? Remembering that every time it comes through ATP synthase, it spins this thing and ATP is created. All right. Again, biochemical pathways are not required, but if you have a basic understanding of what I've just went through, you should really understand what happens in the electron transport chain, the inputs, outputs, and of course the location. Okay. These are your inputs and outputs here. Okay. So again, inputs, we need that oxygen that I mentioned to make that water at the end. All right. Which is why an output is going to be water. Inputs 26 or 28 ADP and inorganic phosphates. And the output is 26 or 28 ATP with my beautiful editing job there. Of course, NADH, 10 molecules, FADH2, 2 molecules is an input. And then lastly, the output there, NAD+, some hydrogens, FAD, and some hydrogens. Okay? So that's your inputs and outputs there. All right, watch this video if you want. Do these questions if you can. All right.